Hello friends, in this video I will explain about the structure and the selection procedure of PBr322 plasmid, and this is the general structure of PBr322 which is present here. You can see it is actually composed of a origin of replication. Here is it is the site where the replication starts, then a ROP gene and a tetracycline resistance gene and a ampicillin resistance genes. These tetracycline resistance genes and the ampicillin resistance genes actually have the cutting site for various restriction endonuclease. As you can see, the PST1 it is present in the ampicillin resistance gene, and the BAM H1 is present in the tetracycline resistance gene, and the plasmid PBr322 is actually 4361 base pair long, and the tetracycline resistance gene actually clone for the transposon TN3, and the ampicillin resistance gene actually clone from the plasmid PMB1. This is the tetracycline, and these are their source. And if I move to the next slide, here how can you insert a your gene of interest within a ampicillin within a PBr322 plasmid, and how can you select the transformed plasmid? Here is the structure of PBr322, and this is your target site. So the first you have to cut both of gene of interest as well as the target site with the same restriction endonuclease. Here is they cut the target DNA into BAM H1, and so that we can insert DNA within the BAM H1 tetracycline resistance gene. So in the second stage, the cut with BAM H1 and then ligate within the tetracycline resistance gene. Here you can see. So if we insert your gene of interest into the tetracycline resistance gene, then definitely it will disrupt the gene function. So we can select based upon this criteria, that if the transformation is occur, then it will disturb the tetracycline resistance capability of the cell. So here there are some petri dishes are present, you can see, and these petri dishes are actually containing agar actually, and with ampicillin, that is the antibiotic, to see whether the plasmid is inserted within the E. coli cell or not. So first the petri dishes are plated with agar and ampicillin to see that whether the plasmid is inserted or not, because the ampicillin resistance gene is intact, but although the tetracycline is not present, because it will disrupt it because of insertion of new gene of interest. So to see that whether the plasmid is inserted or not, we are actually growing the colony with ampicillin, and those colonies are actually present here, the black colors. And these colonies having the transformed E. coli, means the plasmid is present, but to see that whether the transformation is occurred or not, and this there the colony containing recombinant plasmid on ampicillin plate, you can see that this colony, then in the next step the colony is plated with both tetracycline and ampicillin. Because tetracycline gene is disrupted in case of recombinant one, that's why in here those are transformed plasmid are actually unable to survive in the presence of tetracycline. This is all about the transformation of E. coli, to see whether it is transformed or not. Thank you.